It is a keynote, so in this keynote I will try to give a bit of a personal take on all of this. I will tell you my personal journey towards MPS and how it is going at the moment. Before I start, however, I would like to quickly declare a few conflicts of interest. I try to bring the rubber on the ground, and this means also talking and working with the industry. Take everything I say with a grain of salt if you like so. So, the journey from in vivo to in vitro was a journey of simplification, reductionistic, less and less, so that just the key elements were there. And now with MPS we try to move backward. We try to become more in vivo-like. And we cannot do it for all organs, as you can see, but for some organs we can already pretty well. And this is the journey I want to describe. And for me, my journey obviously started at a young age, and three of my passions have been going throughout my life, which is a love for animals. You see me here with my sister Kati, with a young lion cub. Science, already early on, and also politics were my interests. And I, participated, for example, in these science fairs during school, or I did 550 performances of stand-up comedy, political stand-up comedy, so to finance my studies. So this is something which has been going through my life, and I'm continuing to be interested in getting politics right for the science I love so much. I've studied at many places, so you can certainly say I'm broad and not deep. Many scientists have their field. I have many fields I'm interested in. Marcel already referred to my early exposures to animal experiments, and I have to say it was a remarkable coincidence that I had, as a student of medicine, to do an anamnesis. I had an hour time with a 34-year-old woman. It's not she, I just found a picture which was a bit look-alike. We were suffering from liver failure, for children waiting for a possible liver transplant. And the day after, I was witnessing the first animal experiment in my life, in the biochemistry part of my studies. And they were treating for septic liver failure these mice. And I can tell you at the time I said, I would sacrifice any mouse if I could help the woman from the day before. But I never loved it. I always needed a big glass of whiskey in the evening when I was involved in any of these experiments. So I was most excited when I could start my PhD really bench to bench with Marcel. He did the mouse hepatocytes, I did the rat liver hepatocytes. When I could translate this animal model into a PhD thesis, and only towards the end of this thesis, our friend Franz Gruber, who just sadly passed away in April, told me, Thomas, are you aware you're dealing with alternatives to animal testing? And this was actually then getting me into the whole world. And as Marcel already said, for me, some of the lectures of this model, which is a Kupfer cell hepatocyte model, which is still very much in use as a basic model of inflammation, was that we need co-cultures of different cells, and immunocompetence is a very important trait of these systems. However, in my work, I had a tremendous problem. There was one paper from Japan which gave us communication problems, because for 1.5 years I desperately tried to reproduce this paper. I even wrote, at the time it was letters, real typed letters, to these people, never got a return. I don't know whether my letters even arrived. I don't want to blame anybody, but for me it was very interesting, 1.5 years of trying to reproduce something and seeing how many gaps are in our descriptions of doing these things. How many things I had to guess what they had possibly meant when describing their method in a short paragraph. And this got me really into thinking of how can we do better. And it was shortly after my PhD that I got interested in developing good cell culture practices, and in the end this led me on a path towards validation. And also I see in continuation of this the creation of the evidence-based toxicology collaboration, which is a major attempt for improving the quality in the preclinical science. I will come to all of these aspects in a second. Evidence-based toxicology started for us in 2006 when we suggested that we should translate the principles of systematic reviews, of risk of bias analysis, of quality scoring, all of these into the area of toxicology. We organized the first conference in 2007, 170 people from all continents, and since 2009 I have the privilege of the first chair for evidence-based toxicology at Johns Hopkins, and since 2011 we have the evidence-based toxicology collaboration, which is an attempt together with all of our partners to really handle the information more transparently and objectively than we typically do. And here you can see how in our field of environmental health the systematic reviews have been increasing over time, and in these 12 years we have interacted with more than 1,000 people. In our board of trustees we have representatives of the EPA, the FDA, the European Food Safety Authority, and we're really excited how this is taking traction. And we created now the Evidence-Based Toxicology Journal, and we are forming a membership-driven association, so no longer driven by a board of trustees, and I invite you to become part of this. In my journey, I learned more and more that it is not just ethics to replace animals, that there is a matter of quality of science in moving to new types of system. That's a book I really enjoyed. You see the rigor mortis, how sloppy science creates worthless cures, crushes hope, and wastes billions. I'm cited in this book. I gave long interviews for this. You need to publish, and you can't write, I use a terrible model. You have to grant applications. You have to do grant applications, and you want awards, so you only show the good sides. You're penalized if you do it differently, if you're honest about weaknesses. And unfortunately, I think this is the culture in science. I've never read a scientific paper which says, I'm using now a mediocre test, but it's the best I have at hand. I'm waiting for the first one. And part of this is the quality of cell culture. Our traditional cell cultures, as I learned them in the 80s, was pan-fried egg sunny side up. If you give it a closer look, these have a cell density of roughly 0.1% of real tissue. Typically single cell types, no architecture, no functionality, hardly any cell contact. Half of what the cells see is plastic, and the other half is cell culture media. That certainly has to be overcome, and we all know that's why we are here. 3D is an answer to this. But there's also the problem of sudden environmental changes. From 1 second to the other, these cells typically get rid of all of the waste and suddenly have oxygen and nutrients again. This is a shock, yeah? A shock a day keeps differentiation away. It is very simple. Cells have to be extremely adaptable to stand this. Otherwise, they cannot cope with these. And this is something which is, for example, overcome by perfusion. That's why many of us are favoring models of labs on chip. I got interested in perfusion technology very early on. In 91, I was excited to learn about the minute technology, which was a very early commercially available perfusion system, and I started an EU project around this. So we coordinated this project towards standardized liver and kidney perfused cell cultures with this plug-and-play type of system. And we published on this, and this was one of the experiments we did. You see here one of these chambers. These were chips which could be introduced in these chambers, and we did a very simple experiment. We filled the chamber with methylene blue, and then we perfused it. What you can see here, after 0 minutes, 30 minutes, and 90 minutes, there's absolutely no exchange of media taking place between these sheets. So we said, okay, this is not a design which actually works. And the first thing we got as a response was a cease and desist letter to our head of the university. Things can get ugly if you work with commercial entities. We published nevertheless about this system and how to possibly improve it. But this was one of my early experiences with perfusion culture. But not all of them were negative. This is, for example, a paper we did, unfortunately published at the time still in German. it was using this system, and we were addressing a problem which we saw. This is in vivo release of tumor necrosis factor alpha in a mouse or rat system. You see a nice peak after about 1.5 hours. In vitro, the release of TNF alpha is very much delayed, and many of my colleagues said, your systems suck. They do not replicate the in vivo situation. And it took us really the perfusion to understand that in perfusion, we actually saw pretty much the same time course as in vivo. And it's very simple because what you see here is accumulation of TNF in vitro, while it is excreted in the in vivo situation. And if you do the first derivative of this very curve, you see a very nice curve with a maximum around 2 hours. So it is sometimes the perspective is wrong. We are comparing oranges and apples when we are comparing our in vitro setups and the in vivo setups. So it is really important to keep this in mind. Then the next thing, obviously, is organ interactions. Organs are specialized. The body function comes only by the combination of very many of these systems, and we need the immune system, we need metabolism, kinetics are very much impacted by the presence of the various things, and so on and so on. And this is why our increasing developments of body-on-chip systems is the opportunity to replicate a lot of these functions. We are not there everywhere, not in every model, but we are getting better and better to achieving some of this as it is fit for the respective question at hand. But one thing I really learned on this is that bad cells don't get better in sophisticated culture devices. Or measuring complex endpoints like omics technologies or high content imaging. It starts with the quality of the cells. And this is typically overcome with either using primary cells or stem cells. Because we should be honest, the cell culture, the majority of cell culture is a disaster. I call this irreproducibility. About 25% of our cells are misidentified, we have to assume. The last study in 2015 I'm aware of showed for 400 cell lines that roughly 100 of them were misidentified. Yeah? They were not the right species, not the right organ, not the right gender. Yeah? That's our reality. We have to assume that about 25% of our cells are mycoplasma infected in academia. 25%. In some studies, as much as 30-something% have been identified as mycoplasma infected. And I can tell you, a mycoplasma infected cell is not doing a physiological job anymore. But it goes on, genetic instabilities, culture artifacts. Let's talk about the genetic instability. We were shocked in this work in 2016 when we took 2 vials from the same batch from ATCC of an MCF7 cell line. Same vial from the similar vials from next to each other from the same batch. And when we started working with them at Brown University and Johns Hopkins, because we wanted to do direct comparisons, they did grow completely different. They responded completely different to estrogen. We had no idea what is happening, so we started looking into this. And MCF7 cells have given rise to more than 28,000 scientific articles. Yeah? So it's a workhorse of science. It's a breast cell cancer line. What did we see? We did first a karyogram, which was already somewhat surprising. You see some chromosomes could not really be found anymore, others were multiplied. Was a quite interesting thing. And we did then a comparative genome hybridization and found that 10% of the genome was lost. 50% had less than the 2 typical copies, and 30% in comparison was increased up to 30 times. So these are Franken cells. Yeah? They have nothing to do with physiology. And it is pure luck if they perform in any way like a physiological cell. And I can tell you there's similar work done on HeLa cells, and probably most of these cell lines which have been cultured over time are not representative of physiology. Interestingly, in this more recent paper, Alex Mertens and her PhD student in my team showed that they're not even cancer cells. They compared the genome of the MCF7 cells with a broader panel of 1,200 samples of breast cancer cells, and they're completely different. Oops. This is another example. These cell lines were commercially available, pretty expensive in the early 90s, and they were apparently only different in one point, an inserted cytochrome P450 enzyme. What you can see, how many genetic changes had occurred in these, and if you interpret then your experiments, yeah, this is just one enzyme in addition, you will get very funny results, I can tell you. Cell culture artifacts. Our problem is that we want to have a lot of cells and we want them fast. But cell growth is the opposite of differentiation. Very simple. A cell can either multiply or differentiate. We know this from cancer. Cancer de-differentiates and multiplies very quickly. But we do everything to drive them. We give them tons of sugar, growth factors, fetal calf serum. We drive them into a proliferative state, not that we could not keep them slow. We want to work fast. And that's a major problem we are entertaining here. Another problem I see, I described as levels of atrophy, cellular atrophy, because we are boring our cells to death. As you can see here, a muscle which is not in use shrinks. That's what is known as atrophy for physicians, but there's also cellular atrophy. The cells get smaller, they lose function, because the body is highly efficient. We don't maintain capacities which are not needed. Why should we? Why should a cell have a metabolism for exogenous compounds if there is no compound to be metabolized? Why should a brain cell maintain its functionality if it has nothing to think about? And we'll come back to this again. Because I really believe we are boring ourselves to death. We are expecting functionalities while we are not demanding them, and then suddenly in the experiment, the cell should do the trick. And we can continue. Sure, there's good sides of cells. The ethics, for sure. They're human or can be human. The costs are tremendously cheaper than most animal experiments. One cancer bioassay is almost $1 million. Yeah? So if you just compare how many PhD theses we can do for $1 million, it's quite substantial. They're typically faster, so we save time, very important aspects. But we are also spoiling them. We are overfeeding them. We use fetal calf serum with growth factors. We need glucose levels which are highly diabetic. We are using antibiotics which are changing enormously the gene expression just to work not as clean and have not risk infections. But also many other factors are pure artifacts. Why do we culture rodent cells at 37 degrees Celsius? Because we have an incubator at 37 degrees, but that's human body temperature. Mice are 40 to 41 degree. We use very loose pH controls. The human cannot be conscious below 7.35 or above 7.45, but our cell cultures are tremendously erring into both sides when we are working with them. We're keeping them essentially anaerobic. And we see this because our cell culture media are turning yellow. Why so? Because they're only doing glycolysis, and the pyruvate is accumulating and is turning the phenol red yellow. That's certainly not physiologic. So all of this led actually already in 1996 to start something at the time with the German Cell and Tissue Culture Society here in Berlin, the first symposium on good cell culture practice, which I had the privilege to co-organize. And this started a story of developing good cell culture practice, and I already showed you the guidance document we finally published in 2005 by the European Commission, which is a pretty well-cited paper. But 2005 is a year before the induced pluripotent stem cells were even invented. No microphysiological systems around. So we started to team up with a number of organizations and started to do 2 workshops. in which we developed the concept for GCCP 2.0 to extend this now to microphysiological systems. You see some of the organizations which helped us with these things. And 2 workshop reports were the result of this. Good cell culture practice from stem cells and stem cell derived models, and advanced good cell culture practice for human primary stem cell derived and organelle models, as well as microphysiological systems. And I have to mention David Pamiers, who was coordinating these activities for us. And it is really of critical importance to have such guidance, because in the end it is all about quality. And Good Cell Culture Practice 2.0 was published a year ago. This guidance document is now available and is extending to these areas. And we are still working on dissemination strategies, talking to journal publishers, to funding agencies. And I would hope that the Microphysiological Systems Society is going to adopt some of these and is promoting them, because we cannot afford to deliver low quality if we want to be successful with our endeavors. And I could very well imagine that promoting quality standards is key here. And the next step in our quality journey has to be reporting standards. And they're even more important for microphysiological systems, as we are working with highly complex things. We have to find ways of telling others how to reproduce our findings, because there's no gain by saying, I know how to do it, but I don't tell you. We need reproducibility, we need exchange, and I think the society will be very well placed to promote these concepts. These are 2 articles, one spearheaded by Marcel, the other one by me, towards reporting standards and templates here. And I think we are only at the beginning. And also the ongoing work by the NC3Rs is not really yet at the level for microphysiological systems. I think we need to find ways of moving forward that these systems find good reporting standards. A parallel story of my life and work developed, and about 10 years I was working very much around this molecule, GCSF, which is a very interesting cytokine. I don't want to bore you with this information, but for me it had actually 2 very important aspects. One was because of my involvement with clinical trials, I came to the evidence-based approaches of evidence-based medicine, and this is why I could later do this translation. But the other one was I was employing whole blood assays for my volunteers and patients in order to assess their immune function. And when I learned that there's actually a lack of human pyrogen tests, I said, my whole blood assays actually are perfect pyrogen tests. Blood of a volunteer is not releasing any cytokines except there's a pyrogen, like endotoxin. But I also can stimulate them with non-endotoxin pyrogens, so it is detecting things the Limulus assay cannot detect. And I had the idea to develop this assay, and this was a journey of now almost 30 years. 30 years making assays on this principle, now called monocyte activation tests, available. I had the privilege to head the validation study. They became accepted and so on, and we've celebrated the 20th birthday, the 25th birthday of my assay, and similars in the field. And you can ask, how long does it take to get some impact? In 2005, when the assays were validated, we were using in Europe alone 160,000 rabbits. And here you see the actual numbers, the latest from this year, on the year 2020, we are down now to 23,000. So 80% of all animal testing in this field has been reduced. That's a major contribution. If you compare, in 2005, we were using 100,000 animals for all testing of chemicals and 100,000 for all testing of pesticides, and 160,000 for pyrogen testing. It's really an enormous thing. And finally, in 2021, the European Pharmacopoeia declared they will phase out rabbit testing in Europe. No such attempts at the moment. In the US, Brazil has taken this up, but not many other countries. And this shows you that we are often simply lacking implementation. it takes too long. We have no enforcement of the things we already have available. And this, I think, is a very sad story we have to tell. It was the successful coordination of this validation study which gave me the opportunity then to join the European Commission and get responsible for validation studies. This is the beautiful Lago Maggiore, and I can't wait, I tell you, tomorrow evening to go there and stay for the summer again. It was a very interesting time. We were well equipped, and with enough money you can achieve something. The validation studies we started in these 6.5 years I was there led to more than 20 OECD test guidelines and monographs based on these validation studies. If you put money in, you get something out. So this was a very important validation experience. But why do we need validation at all? Because we love our ugly babies. You will never turn against your essays. You need somebody independent to look at the weaknesses of your essays. We are as scientists not very self-critical. We are very critical of our competition, but we are not critical of ourselves. And this is why we need this independent process, and we cannot expect the regulators to do this either, because they have to handle far too many things, and they cannot have the overview about each and every methodology. But validation is roughly 1 million per test, takes you 10 years, and 40% of them fail, despite the fact we put our money only on the best horses. So it means killing meticulously half of your scientific children. This is why it is not very popular. But if you now come to MPS, good luck in validating something like this. Many of these systems do not even exist in other laboratories. So we really need to find new ways of handling this enormous number of variables. There are also moving targets as technologies which we are changing throughout. The costs and the throughput of this system does not really allow large validation studies easily. And we often have no reference point. There's not an animal experiment we can use to compare to. Very often there are systems in their own right. And we also don't want really to freeze them in time. A validation study, 10 years, 1 million, probably much more for complex MPS. Do you want to freeze it in time? No, you want to have the next generation, the next advancement next year. And then you have to, in principle, restart again. There's ideas on how to do things better. I'm just putting out here two. We have been developing a lot of the evidence-based toxicology with the idea, can we use these principles in order to validate systems in a different way? A very important thing which is now being taken up increasingly is the aspect of mechanistic validation, saying the thing which counts is, are the mechanisms, the pathophysiology, is this reflected in our test system? That's what we have to show, because then we can compare to humans and our knowledge about this, and not a Fisher table on do we have a congruence of positives and negatives. We also have to move finally out of this dance around the golden rat. We have to find ways of really humanizing these things. And I don't enter today into something which has made me a living. I'm not beating a dead horse, talking about the shortcoming of animal tests. This is closed, we know it. About mice and rats predict each other 60% of the time. We did a big study with about 3,000 chemicals in 9 commonly used animal tests, and we found 81% reproducibility of these animal tests. These are GLP done OECD guideline tests, the best you can get. Simple high-dose tests. And the numbers are still inflated. For toxic substance was only 69% reproducible. I would not sleep well as a regulator if I knew that my systems are only finding 69% of toxic substances. There's also pressure mounting. We should be aware that society expects us to replace animal testing. About half of Americans, up by about 15% over the last 20 years. 60% of Europeans don't want animal testing for medical tests. For cosmetics, we are in the 70s, 80s, 90s. Yeah? So we have to simply see a general population wants us to change, and politicians here, they listen. And we see on both sides of the Atlantic increasingly legislation, commitments in order to phase out animal testing or reduce it at least dramatically. The most recent one was December 23rd, when President Biden did sign the FDA Modernization Act. And this is something we all should cheer. We should be celebrating, because this is the first time, to the best of my knowledge, that microphysiological systems made it into legislation. It says here explicitly defining non-clinical tests for the purpose of FDA with microphysiological systems such as organ chips or bioprinted or computer models, it says. Yeah? So this is we have arrived, we are there. We should also say that this is a highly symbolic act, because it does not change anything in the procedures of FDA. They would have accepted these information sources also before, but nobody dared to submit these things. So it is highly symbolic, and that's good, because it tells the FDA to rethink, it tells the submitters to rethink, and it tells the biotechnology industry there is a gold rush possibly coming, because things are changing. And this is of critical importance, not only for us in the US, it is of critical importance overall in the world. And why so? Because of the global pharmaceutical market. We are just 4% of the world population, but we represent 48% of the drug market in the world, and 64% of the drug market of things under patent. And this is why the FDA is more equal than others. And this is why it is so important when you hear Susie Fitzpatrick, the host of last year's MPS conference, or Donna Mendrick here talk about these systems and the willingness to promote them. This is open doors, yeah? And this is of very, very critical importance for our field to move forward. And there's a lot, from my point of view, broken in the pharmaceutical processes. It takes you 12 years to develop a drug on average. The costs have been calculated at 2.4 billion to come there, but if you also factor in the failed studies, Forbes has already in 2012 said it's about 4 to 11 billion to get one successful product on the market. So which means that one day delay in registration is worth several millions, because they have to make the money back in just 8 years until the patent expires. This is, if you could shorten this with any type of test system, if you can predict any better what the outcome will be, to reduce this failure rate, which Aerosmith has calculated 95% of everything going into clinical trials, a lot of this because of toxicity, for example, would be something. But also, after all of our processes, 1 in 100 patients dies from drug interactions which were not foreseen just by being hospitalized. 1 in 100, yeah? And about 8% of all drugs are later withdrawn, essentially because of side effects, which were not found even in our large clinical trials. So there is an economic imperative in order to move forward with our systems. And this is one of my most downloaded articles, which is the most important omics is economics. And I think it is really important to understand that we are making progress. Interestingly, I always saw toxicology as remaining far too long in animal science. Markets and Markets analyzed that it is 4.2 billion we spent on animal-based toxicology, but it is 14.4 billion already for in vitro-based toxicology. And we have good data from the EU, which show very clearly, while pharmaceutical companies were increasing their research spending between 2005 and 2011, they reduced animal use by 40%. So there's clearly a trend in this industry which is really under enormous pressures to deliver as fast and with the best quality as possible. And if I stay with the toxicology side, we have been working with a group of European investigative toxicology leaders in the pharmaceutical industry carrying out this workshop. And what we saw is there is now actually a toxicology beside this black box here of regulatory toxicology, because we have both a prospective and a retrospective toxicology. A prospective one which is trying to already identify is the target associated with any type of risk, to de-risk before doing regulatory testing. And a lot of the work is spent actually to de-risk, to have an animal test result and then to show it is not relevant for humans. This is really water on my mills, that a lot of the work in the industry is spent to show that animal tests are not relevant. And a lot of money is spent here. And I put some numbers behind this. These are only my personal educated guesses, but I would say that a lot of de-risking is done with non-animal methods, while we still have not really arrived in replacing regulatory testing with a lot of these non-animal methods. But we should be clear, for us as an MPS community, the markets are outside of the regulatory testing at the moment. This is where things can be done and where a lot of our turnover is being done. Another driver which is really critical to keep in mind is that the type of drugs is changing. Nowadays, about half of the drugs are not synthesized chemicals, but they are human proteins or antibodies against human proteins. They are very species specific, and this means also that animals are useless. And this is why we need human test systems. And in the absence of good human cell culture, the MPS and stem cell derived systems are the solution for a large part of these. Good. I took over in 2009 the Center for Alternatives to Animal Testing, and yeah, this was very much the situation of animal testing, then a miracle occurs and we arrive at toxicology in the 21st century and no animal use. What we really had to do is develop strategies, because I think it is about a roadmap to get out of the current dilemma to something different. And actually I was given the opportunity to develop with a group of about 30 avant-gardistic toxicologists, those who are really moving ahead of others. And we developed a plan how to do this. I found with Ana Navas-Asian from Colombia and with Waiche Shu from Texas A&M, two co-chairs, and with 15 agency observers, we published 3 weeks ago this document for the Department of Defense, where we call for a human exposome project. And it says, and this is an article of mine which is summarizing some of this and also talking about the downstream activities. And we say we need to make toxicology more exposure driven. We need to embrace technologies like MPS and artificial intelligence, and we need to find ways of integrating evidence. And I will not go into too much detail, but I believe very much that a big part is not to sell the link from a chemical to a hazard in a rat, but the link of a chemical to disease. Because we have learned after 40 years of the Human Genome Project that 5% of diseases are genetic and 40% have a genetic component, but there's 70 to 90% of diseases which have an exposure component. So it will be very important to start cataloging this. And here come microphysiological systems, and I don't dare to tell you what they are and what they should be. But we have done several workshops on the topic here in Berlin. Marcel mentioned them. Adrian Roth and Marcel and Uwe Marx were spearheading these. And we have documented the enormous progress our field is making. The last workshop led also to this science paper, which we published in 2021, which is a summary of the second workshop, and I invite you to read this. And we just had the third one of these as a pre-conference workshop, and we found again a lot of interesting topics. But another offspring of the second workshop was that over a beer, Dan Tagle and myself discussed we need a society, we need a series of international conferences on this topic, and that's why we all are here. And I thank Dan that he implemented this and found a way to get NCATS to get us started on this on this journey. We found the first workshop hosted with Don Ingber and Susie Fitzpatrick of the FDA in New Orleans. You have the hosts here of this workshop, but more importantly, there are people who actually did the job. And I would say, I would single out among the many people who have done work. We have enormous groups, but I still think we have to single out these three in our team. the tireless work of Lena, Camilla, and Anwin needs to be acknowledged. And since I don't know whether I have any opportunity to say it later, I say it now. Our field is developing at the moment to something which I like to call micropathophysiological systems, because we are able to more and more model diseases. Our own interest was prompted by the fact that autism is rising with an enormous velocity. The data from March this year say 1 in 36 children in the US is diagnosed with autism. And you see the enormous increase, which in latest year can no longer be explained by higher awareness and better diagnostics. We really have to ask, is there developmental neurotoxicity of chemicals involved? Because genetics don't change. And quite interestingly, we worked on this since 2005. We did 9 workshops, 4 international conferences. The 5th one is planned now for 2024 in Constance, hosted by Marcel and Ellen Fritsche, who's also somewhere here at the conference. And this was driving us to start targeted development of tests. And the big news is this year we will get an OECD guidance document on in vitro development and neurotoxicity testing, which is the direct result of these 17 years of collaboration and targeted development. And we did choose out of this to develop brain organoids, which we announced in 2016. David Pamias is here, was leading this as a postdoc and later research associate with us, Helena Hochberg, and many, many others who were involved. I don't go into any level of detail. These are the first mass-produced standardized brain organoids, the 4th group which was producing brain organoids at the time. You see the delay from 2006 when stem cells became available to 2016 to get something here. And under Lena's lead, we have been using them for many applications, and you have heard some talks. Many of our students are presenting here to use them using different genetic backgrounds, manipulating them, introducing risk genes, repairing them, reporter genes, and so on and so on. We have very nice myelinization. We can add microglia to make them immune competent, and we can do a variety of disease models, gene environment studies, and I will quickly show you, just for the flavor, a few of the things which have been done most recently. This was one which I find really remarkable, because here, for the first time, the combination of a risk gene and a risk chemical for neurodevelopmental disorders has been shown. CHAD8 is known to be a risk factor to develop autism, and chlorpyrifos has been banned in the US just around the time of this paper because of its possible liabilities here. And in their work, they showed this synergy, and this led now to a very large size clinical study, epidemiological study. 18 centers are following 175,000 children to find such combinations, and we are tasked to demonstrate then in vitro in our brain organoids whether there is actually such a synergy between the assumed pairs. Another work which also shows how synthetic biology is there to enhance these systems beyond what we can do in vitro. With CRISPR-Cas, Lena and team have been introducing in this paper, for example, a marker for oligodendrocytes and one for synapse formation. So non-invasively, it allows to follow very nicely the development, the ordinary or not ordinary development of myelination and synapses. And this is just starting to really develop a mini brainbow, as they call it, where they want to follow the different lineages in just one microphysiological system. Other work sponsored by the FDA and by NINS is addressing mixture toxicology, which is somehow the holy grail. And Lena and Fenner in our team are spearheading these two activities, which are around heavy metal mixtures, very obviously involved in neurodevelopment and neurotoxicity. The autism numbers, which I showed earlier, are shown here in a different way, and I use this just to show you something which I found quite remarkable, which is that we have lately an interesting increase because it seems to accelerate. There was linear increases over long periods of time, and suddenly we see more autism. And I was wondering, could this be linked to the pandemic? Because we were interested very early on, can the brain possibly be infected with SARS-CoV-2? And we found partners with Bill Bissai and Corinne Gwillen And we showed in May of 2020 already that we can actually infect human neurons with this virus. This was at the time the first description that this virus is actually infecting human brain tissue. Led to a lot of interest. Financial Times and 400 others reported on it. We published this peer-reviewed in June of 2020. And if you imagine, it shows you how fast these enabling technologies are available for an emergency type of assessment. In the meantime, more than 10 groups have replicated these findings. They're also in line with the clinical findings and shows that a human test system can show something which actually is difficult to do otherwise. And in general, if there's something positive about the pandemic, it is that it has shown to us that these in vitro tools can play an enormous role, or that the other way around, we don't really need the animal studies to develop good drugs. Normally, 6% of vaccine projects lead to success. After 10.7 years on average, we found 9 vaccines within 1 year successfully doing the job. We developed, not we, the scientific community, the pharmaceutical industry developed 2 antivirals within 2 years. And this was all possible because we did skip a lot of the normal development steps which are done in the variety of models, including animal tests, which is a lengthy part of it. And if you look into the systems, the human lung is infected by the virus, we know, but we also had to learn that all of the other organs are getting infected. Animal models, rodents cannot be infected at all. Hamster and ferret can, but it's a very mild disease. They're certainly not dying from it. And monkeys, macaques, they reasonably show this, some limits, but also not organ contributions. But the human microphysiological system, they do the trick. There's a rich literature showing already in 2020, the same year, for essentially all of the organs that MPS are suitable to help us with these type of diseases. There's a lot of enabling things happening. For example, we can start freezing organoids. These are 2 brain organoids. One is the control, the other one was frozen and thawed. And you can imagine what this means. You can send them, you can store them, you can have them available. Oh, today, not today, but I will need for my experiment 3 Alzheimer, 2 ALS, and whatever. And you can choose the gender. You have them all in your fridge. And you might use them at places you not typically think of. This is work which was just completed and we will present at the cryobiology conference for the first time, is showing that these are actually electrophysiologically more active than the non-frozen ones. So I'm already thinking about freezing my brain for a while. It could be actually helpful. But it is showing that you can combine our technologies with other disruptive technologies. And this is an enormous opportunity. If you mix different disruptive technologies, you get enormous synergies. And I want to show you only one we are very interested in, which is the combination of our brain organoids with artificial intelligence. I'm field chief editor of Frontiers in Artificial Intelligence because that's my other leg of working on AI. It's an area where we all witness enormous progress because the capacity of AI is doubling every 3 months over the last 10 years, which means per year about a 10-fold increase. And that's what you can see with the large language models, the generative AI, and name it. Also, the data are increasing by about 60%, and the computer capacities are doubling. So, which means there is an interesting question now: what can AI do with an organoid? And we call this OI, organoid intelligence. So the idea is that we can do a synthetic biological AI, and instead of making the computers more brain-like, what we have been doing throughout the history of computer, we are making a brain culture more computer-like. And this is interesting, even though I'm comparing apples and oranges. If you see, this is the best supercomputer in the world, the Frontier computer in Kentucky, and only last year in June, exactly 1 year ago, it reached the capacity of a single human brain at 1 exaflop. Yeah? This is a $600 million installation with 680 square meters. So So this is really something. And it is using about 1 million times more energy. Or you can compare it instead with a laptop which has the weight of your brain, but would take us another 33 years to make our laptops as powerful as a human brain, if we can continue with Moore's law. Yeah. So there's an enormous potential here. And we were triggered by the fact that our cells were spontaneously electrophysiologically active. You can see here these calcium fluxes, which I found really impressive, watching the brain think. And when some people were shocked, I said, yeah, they're thinking, but they have nothing to think about without input and output. So the idea of organoid intelligence was to give them input and output and feedback on what they're doing. And we believe that this organoid intelligence will allow us to do basic physiology of learning in a human experimental system, will allow us to study brain-machine interfaces, allow toxicological testing and drug development, and might also give us some ideas for biological computing. We are on the way. This is a study which we published with colleagues in Hopkins in Science Advances in August last year, where we created an EEG around the brain organoids. So these are self-folding shelf electrodes, and you can study then the effect of substances on the EEG in this. This is not completely new. I hope we can start this. Okay, if it would run, you would see here work by Steve Potter, who controlled with rat 2D cultures already 2001 this little robot, which was moving around. Colleagues of ours who are part of our organoid intelligence initiative from Cortical Labs in Australia, they had a very nice publication where they showed that neural cultures can play the computer game Pong. And again, it runs, yeah. You see here, this is the Pong game, which is controlled by neurons, and they get better in the training session. They learn how to move the pedal more effectively. Lena and myself had the possibility to publish an editorial with the article which came out, and they are now using our brain organoids because the hope is that we also will achieve long-term learning with these type of setups. And the enormous potential of an organoid here is really the higher density, the fact that myelination is present, which is increasing conductivity, more synapses, glial cells are present, which can prune the synapses, which is the way we understand it is working to get the long-term memory established. And you see Alison Motry here, also a partner in crime. He has been, it does not, okay. The video doesn't play. You see him with his robot, and this robot is actually controlled through Wi-Fi by brain organoids in an incubator, and he claims it has learned walking. It's not published in peer-reviewed literature, but it is moving forward. So these systems are at the moment being scaled, while our 500 micrometer organoids have just the number of brain cells of a housefly. This is a 1 centimeter brain organoid. This is already twice the size of a mouse brain, so you can expect to reach something here. In our ongoing work, we are increasing the number of oligodendrocytes and astrocytes to physiological level. We are adding microglia, so we really have the machinery present here. This is all combined with a lot of ethical work. We have many ethicists included. They go with us, plan our experiments, they observe us in the lab, because we are aware that this is raising some questions which we have to address before we run into possible consciousness or anything. But as much as AI depends on the input data, that the answer is in the data, the brain cannot be expected to show anything like emotions or suffering if no such input is there. But it's also an enormous opportunity for experimental ethics and philosophy. When is the setup sufficient to claim this is intelligent, this is aware, or this conscious? We are privileged to have many partners in Hopkins and are being funded by Johns Hopkins with their Surpass program. We are working with high-density electrode arrays in order to carry out experiments on this. There's posters here on this topic, and we are trying to learn to teach these systems and to establish that the connectivity is changing in response to learning. This has obvious opportunities for drug development. Think of dementia, Alzheimer, but also neurodevelopmental disorders, side effects of drugs. We are working, for example, with our partners in the Alzheimer Institute in Hopkins. They have already shown that cells from Alzheimer patients show some of the hallmarks of Alzheimer, and they allow us now to develop these microphathophysiological systems. We are working on what we call the Dementia OI Research Initiative. Other projects are linking us to ALS, amyotrophic lateral sclerosis, autism or leukodystrophies, all diseases for which we are developing the brain organoids while developing organoid intelligence as an output. And I think this is a trend towards hyperphysiological systems. We are no longer limited by replicating human biology. We can produce a 10-kilogram brain if we want to. We can enter through synthetic biology whatever functionality we want. We can form and combine units of various different functions. I think we are only starting to really see how synthetic biology is allowing us to synthesis. This is all science fiction at the moment, and so was a brain-based computer in 1966 when in Star Trek Captain Kirk found a computer based on human brains. So again, there's nothing new under the sun. Organoid intelligence for computing had already been foreshadowed. A lot of this has been published on 28th of February. Kid versions, lay people versions, a lot of editorials. The news release led to more than 600 press hits. There's a lot happening here. A workshop report from Forming a Community was published with a Baltimore Declaration towards OI. All of this available. And we are following the role model of AI here, which was created with a workshop and Forming a Community. And we have this first community workshop. We're trying to find philanthropic money through the Whiting School of Engineering for an organoid intelligence call center, created a journal as a sub-journal of Frontiers in AI, on Frontiers in OI. We just received 2 days ago the first impact factor for the journal of 4, so we're quite happy to start. And we have a very big team of about 80 researchers which are involved with us. So my last slide is that I believe we are on a tremendous journey towards MPS. We see that we have more and more the tools available, and it's all about change management. We should much more discuss on how we can get these things into the use they deserve. It is no longer really to worry about development and their capacities. So enjoy being part of the rise of MPS. And I like to quote Victor Hugo, "Nothing is more powerful than an idea whose time has come." I think the idea of MPS has come. Thanks a lot.